I'd like to thank the organisers for inviting me, and today I'd like to introduce you to some work that we've been carrying out at KaloBios. Hopefully you've heard of KaloBios. KaloBios is a publicly quoted company. We went public at the beginning of this year, and we're developing monoclonal antibodies. And these are first-in-class programs against targets that others haven't either taken to the clinic, or at least we're probably ahead of them. And we're really focusing on serious respiratory diseases and cancer. Cancer is an obvious one for monoclonal antibodies, but maybe not respiratory diseases. And there's all questions about whether monoclonal antibodies can actually get to where they need to get to treat respiratory diseases. As you'll see, our first program, KV001A, is for the treatment of Pseudomonas infections in cystic fibrosis and in ventilator-associated pneumonia. One of those is an acute indication, ventilator-associated pneumonia. If you're on a ventilator in a hospital, you have a tube down your throat, then if you get a Pseudomonas infection, it colonizes the tube, and then it seeds into the lungs, and eventually you develop pneumonia. And Pseudomonas pneumonia has a very high mortality, not really the thing that you want. This antibody actually prevents the development of pneumonia. Cystic fibrosis patients are chronically infected. By the age of 20, 80% of cystic fibrosis patients are chronically infected with Pseudomonas aeruginosa. It causes rampant inflammation in their lungs and causes lung destruction. Those individuals tend to die in their early 30s and 40s. So two very different indications. One is being developed by our partner, Sanofi, that's a VAP indication. KaloBios are developing the cystic fibrosis indication. KBO3 is an anti-Gm-Csf antibody for the treatment of severe asthma. In this case, we're not talking about people who take puffs every so often when they can't breathe. We're talking about severe asthmatics who really can't walk around the block. And they have really very, very severe inflammation, and we believe that that's driven by Gm-Csf. And then finally, we're working with a very novel antibody, KB004, which is an anti-FA3. FA3 is a receptor tyrosine kinase, and this is for the treatment initially of leukemias, and later on we think for solid tumors. But first of all, the first part of developing a monoclonal antibody, of course, is to make it non-immunogenic. And the technology that we use in this case is a process called Humaneering. We can start with an antibody from any source. It can be a human antibody that aggregates and precipitates, or it can be a mouse antibody or a rabbit antibody. And basically, we've been able to identify within the CDR3, and these are sequences that form a pair. They basically are important for the specificity of the antibody, not necessarily the affinity, but the specificity. And each of our antibodies has got actually quite unique properties, and it requires them to stay on epitope when we engineer them. And then we take those minimum essential binding elements, and we put them into a library of human V segments. So basically, every member of our library binds to exactly the same epitope. So we don't need 10 to the 13 antibodies to screen. We only need 1,000 or 10,000 to screen, because everyone is binding to the epitope of interest, but they all have different properties. And really, this is where we come to look at further downstream processing and developing of the antibody. We want an antibody that behaves well. We want an antibody that may have a specific on rate or off rate. And we certainly don't want antibodies that aggregate and are immunogenic. And so within that library, we can select the properties that we need for our antibodies, because they all bind to exactly the same epitope. They're all on specificity, but now we can pick other parameters. We do all this in bacteria, but we actually use a system which is called a signalless secretion system. So there's no signal sequences on our variable regions, and we believe again that signal sequences probably select for what actually gets secreted from E. coli systems. And we know, for example, mouse variable regions are very poor secretors in microbial systems. But when you take the signal off, you find that you can actually get secretion of probably most of the V regions. So you can actually screen them. It's difficult to screen V regions if you don't secrete them. And at the end, we end up with what we call a humaneered antibody, which if you look at the sequence of that antibody, in the V segment in particular, it's over 90% identical to human germline. So these antibodies, I always say, are more human than human. You take a human antibody, or you take a human antibody that's been highly mutated, it's less close to human germline than our humaneered antibodies. So this basically just shows schematically how that process works, identification of the minimal essential binding elements, and then we create our library of human antibodies that we can screen. We can also add diversity by picking those initial binders to increase affinity, by then changing the front ends and the middles and making a combinatorial library, again, of antibodies that all bind to the same epitope. In that way, we can increase diversity, we can increase affinity, and we can also increase these other properties that we're interested in and that we can screen for. I've talked about this. So now thinking about how we actually do this, it's very, very simple. So it's all done in bacteria. We plate out our library on an agar plate that's a non-expressing system. Again, a lot of people will have their expression vector on constitutively, and that will lead to all sorts of selection. Bacteria don't like to be making large amounts of protein while they're growing. We grow our colonies up non-expressing, and then we transfer those to a nitrocellulose membrane, and then we place that membrane on the top of a second membrane, and that second membrane is impregnated with our antigen of interest. Now that means that the antigen of interest can be a protein, it can be a complex mixture, it can be a cell wall, it can be a carbohydrate. It doesn't really matter as long as it will stay on the membrane. And then we put those two membranes down on agar plates that contain IPTG, the inducer of the antibody. And the antibody fragments, the antibody is induced as a Fab fragment, so we can look at single site binding. And the fragments are basically secreted by the bacteria, pass through the first membrane, get to the second membrane, and they either bind or not to that lower membrane, to the antigen on the lower membrane. We can then take that lower membrane and we can wash it for long periods of time. We can wash it in salt, high salt, different conditions, and we can look for which of those antibody Fab fragments remain bound. So we can select, therefore, for faster on rates and slower off rates and other properties in terms of stability of interaction with the antigen. So on the right-hand side of the slide, you can see a developed filter, and you can see in different intensities of spots. And we've spent ages arguing between ourselves about, is a good antibody a big spot, or is it a very tight spot, because the antibody is binding to the antigen and not diffusing. Unfortunately, for each program it's different, so we can't really look at that filter, but we can subject the filter to different conditions and look which colonies have secrete antibodies that stay on or elute quickly. However, be careful what you ask for, and I think this has already come up. If you look at antibodies that bind very strongly, sometimes you actually select for exactly the thing that you don't want. So in this particular slide, in the bottom panel, you can see an antibody that stays on in a 40 bio assay. It looks as though it binds really well, and another antibody that has a faster off rate. In fact, the antibody that stays on, you can see from the HPLC trace, is the antibody that aggregates. So again, you can screen these out, and I can tell you it's a lot easier to screen out these V regions that behave badly at the beginning of your therapeutic program than to wait until you're paying millions of dollars to scale up the antibody production and you're told you have a formulation issue because your antibody keeps aggregating. Now, most antibodies have the same constant regions. They vary from V region to V region because that's where your specificity comes from. Unfortunately, that's also where your problems come from when you start scaling up the production. So good to look now and select your antibody V regions now, and this technology will give you hundreds V regions, all binding to your epitope of interest. So it allows you to screen for those. So these are some of the properties of humaneered antibodies which we think are important. Obviously, the V region sequence can determine immunogenicity, although again I would point out you can take a human protein and you can make it immunogenic in humans. People have done it with EPO and other proteins. They're not engineered antibodies, but if you actually mistreat a human antibody or aggregate it, then you can make it immunogenic. So the primary sequence is only one element of what determines immunogenicity. Affinity, you can select both your on rate and off rate. Most people want the highest affinity. Most people want sub-nanomolar these days. Expression level, we're all very conscious of can you produce these antibodies. Everybody these days, again, wants grams per litre. The antibodies that we've generated, we can generate 2 to 3 grams per litre in CHO cells. And then the physicochemical properties in terms of formulation. We don't want aggregation, we don't want stability issues. Okay, so the difference between a humaneered antibody and antibodies that are either CDR grafted or even fully human is that we have many more of those less of those yellow bars in those V segments. So we're much closer to human germline. And therefore we think we have a greater chance of being less immunogenic. So the question is the proof of the pudding of course always in the eating, and we've taken 3 of these antibodies now into the clinic, and these indications have not been indications where the patients are grossly immunosuppressed, and therefore some of these have got quite competent immune systems. KB001A is an antibody against Pseudomonas aeruginosa PCRV protein. PCRV sits at the top of a spike called the type 3 secretion system in Pseudomonas, and it's involved in allowing the secretion of cytotoxins. What Pseudomonas tends to do is it tends to inject these cytotoxins into epithelial cells and also into immune cells. It's one way in which it evades the immune system. It's also a way in which it creates inflammation, which also causes damage to the epithelium and allows the Pseudomonas to invade the soft tissue. We've generated this molecule as a pegylated Fab fragment, and you may say, why have you done that? Why not have a full antibody? The reason for that is that in cystic fibrosis lung, there's already rampant inflammation. We really don't want to be making immune complexes with many FCs between the Pseudomonas and the antibody fragment. And therefore, we created a Fab fragment that was pegylated. In fact, the activity of this antibody is that it binds to the PCRV protein and it blocks that secretion mechanism. So it actually blocks secretion of the toxins, and it has to be at a particular epitope on the type 3 system. We also pegylated it because in the lungs of CF patients there's lots of neutrophil elastase. In fact, antibodies don't last very long in the cystic fibrosis lung. This antibody has a half-life in the blood of 10 to 14 days and seems to have a similar residence time in the cystic fibrosis lung. So this is an antibody that's been optimized beyond just humaneering. The second antibody, KB003, is a human antibody against Gm-Csf. It's the human IgG1, relatively straightforward antibody in this case, but it has to be high affinity in order to block the Gm-Csf activity. So this antibody is a 20 picomolar antibody. That's what it was 30 picomolar as a mouse antibody, 20 picomolar as a humaneered antibody. And also, KB003 has a very long half-life, 21 to 24 days. And for chronic indications, we like indications where we don't have to frequently dose. One other property of this antibody is that it's highly soluble. So it's actually soluble up to 250 milligrams per mil, and we have stability over a year at that concentration. So again, tells you the importance of picking the right V regions when you're making these antibodies. KB004, which is our final program, is also in the clinic. this antibody is a non-fucosylated antibody, so it's optimized for ADCC, but it's also a very, very interesting antibody in that it binds and it triggers the FA3 receptor. And it binds to a conformational epitope on the FA3. And so when we humaneered that antibody, we really had to stay on epitope. And when it activates the FA3 receptor, it's actually binding close to the ligand binding site, but it's not binding to the natural ligand binding site. And instead of triggering shape change, which is what FA3 is normally involved in, it triggers apoptosis. So this antibody will directly kill any cell that has FA3 on the surface. And we found that leukemic cells and the stromal cells that surround those and the neovasculature express FA3. And we can actually infarct those cells, we can kill those cells just by giving the antibody on its own. But we know that tumor cells are much more difficult to kill in a patient than they are in a test tube. So we've actually given this antibody increased ADCC and also this ability to trigger apoptosis directly. So just to say a little bit more about KB001A, what is the experience with KB001A? We have actually completed several clinical trials already, and we know it has some of the properties that I mentioned earlier. So just to remind you, this is a cartoon of what KB001A does. Here's a Pseudomonas. It punctures holes in adjacent cells. It secretes its toxins. And the antibody has to bind right at the tip in order to block that. Now, the toxins not only kill cells, but they also cause release of pro-inflammatory cytokines. So by blocking that tube, you block both the inflammation and the cytotoxicity. So what's our experience in the clinic with this molecule? We did a single-dose safety study in healthy volunteers, 1, 3, and 10 mg per kg, and we saw no immunogenicity with this antibody. We then carried out 2 studies, one in mechanically ventilated patients at doses of 3 and 10 mg per kg. Again, saw evidence of activity and protection and saw no immunogenicity. And then in cystic fibrosis, we did a study also at 3 and 10 mg per kg in 27 patients, and again saw no immunogenicity. Currently, this molecule is in a multiple dose phase 2 study that's ongoing. So just to show you that this antibody really does work, so this is looking at what we've listed here as PA event-free survival. So ventilator-associated pneumonia is a little bit like septic shock. These patients become Pseudomonas positive, and they're either alive or dead in day 28. So in this case, patients were screened, their endotracheal aspirate was screened for the presence of Pseudomonas. If they had Pseudomonas, they were entered into the trial, and then they were randomized either to no antibody or either to 3 mg per kg or 10 mg per kg, and then they were followed for 28 days. And you can see that 80% of those patients developed some form of Pseudomonas event if they were on placebo. But those patients who were treated at 10 mg per kg single dose with a pegylated antibody fragment, 40% of those, 46% in fact, had no PA event. And in cystic fibrosis, we had a different type of measure. In this case, we were measuring inflammatory cytokines in the sputum of the cystic fibrosis patients. As I mentioned, neutrophil elastase is one of the key enzymes that are involved in destruction of the lung of cystic fibrosis patients. And in a single-dose study, again, we saw a nice dose response in reduction of neutrophil elastase within the sputum, within the lungs of the cystic fibrosis patients. So not only are these antibodies appear not to be immunogenic in this setting, but they also seem to have activity. So moving on to the anti-Gm-Csf program. Gm-Csf itself is a pro-inflammatory cytokine and has been associated with a number of different diseases, autoimmune diseases, and we have carried out a number of clinical programs both with KB002, which is the chimeric precursor of KB003. And we're just in the middle or towards the end of a program with KB03. So this is one of the few cases where we've taken the chimeric version of the antibody to the clinic and looked at immunogenicity, and then we've taken the engineered antibody to the clinic and also looked at immunogenicity. So this is the real test of, does this technology work? Is the first antibody immunogenic or not? And so the answer is KB002, which is a classic chimeric, mouse variable region, human constant regions. This antibody was looked at in healthy volunteers, in rheumatoid arthritis patients, and also in asthma patients. Around 59 to 60% of those patients raised antibodies against a single dose of the chimeric. We've now carried out single-dose healthy volunteer study with KB03, and also a multiple-dose running study in patients. That was 5 doses at 600 milligrams of KB03. In none of those studies did we see any immune response against KB03. So I think this is a true test of have we actually de-immunized, if you like, for another want of another term. Have we actually taken away the immunogenicity? It does look with this antibody that the Humaneering has created an antibody which is not immunogenic in humans. Currently, that antibody is undergoing 160 patients repeat dose study. It's a 6-month study in severe asthmatics. In fact, we've just completed the dosing. Results should be coming out in February of next year. What does the KBO2 do in patients with severe asthma? And this is also quite interesting. Because what we did see is we saw a big increase in FEV1 in these patients. This really is a measure of doing something in the lungs. So we saw an improvement in FEV1, but also we assayed the aspirates from those patients' lungs, and the patients who improved in terms of their FEV1 also had antibody in the lungs, and the patients who didn't improve didn't have antibody in the lungs. So again, it's not cause and effect, but it's an association. Okay, moving on to the final program, KBO4. KBO4, very interesting molecule. As I said, this antibody triggers apoptosis directly in primary leukemic cells. And we've recently just established a screen whereby we can screen patient samples for whether or not their tumors express FA3. And we're introducing that in our phase 2 study, which we're about to start. We haven't quite completed the dose escalation because, like all antibodies, even though this is a non-fucosylated antibody, there's no toxicity in humans. So let me just move to this slide. So this is our dose escalation study. So this is total dose per week of a non-fucosylated antibody. It's very, very well tolerated. And to date, we haven't seen immunogenicity with this antibody either. We have seen some indications of activity. Why might we see that? We might see it because this antibody directly kills primary leukemic cells. This shows a nice dose response of the antibody with primary leukemic cells. What's interesting with this is we can kill primary leukemic cells at a dose that's very easy to achieve in humans, at a dose between 1 and 10 micrograms per mil. Normally we think of maybe 20 or 30 micrograms per mil. So this is these primary cells are very, very sensitive to killing with this antibody. But what is really exciting is where the antigen is expressed. It's expressed on the tumor stem cells and on the stromal cells that provide that stem cell niche and the new blood vessels. And so what we feel is that we can either kill the tumor cells directly by this triggering of apoptosis, or we can kill it by ADCC because it's a non-fucosylated antibody, or at least we can disrupt the stem cell niche, and then we can combine it with chemotherapy. This should give us a very big therapeutic window because of the tolerability of the antibody. And I think in the future, people are looking to combine antibodies with other agents therapeutically because they tend to have a very good safety profile. So in conclusion, Yes, we've generated antibodies which we believe are not immunogenic. We believe they're not immunogenic not just because of their variable region sequence, but also their physical chemical properties. We can take nice human proteins and make them immunogenic. We can take nice human antibodies and probably make them immunogenic, but we need to be able to not over-engineer that variable region so that those antibodies start to aggregate or misbehave and become immunogenic for other reasons than their primary sequence. Our antibodies have been dosed both in single-dose and multiple-dose studies. We have yet to see immunogenicity. Will we see immunogenicity? Surely. If we treat enough patients, I do believe most things are going to be immunogenic. But clearly these antibodies have very, very low immunogenicity. But they are all each different. They've all been optimized to either to kill tumor cells, to clear cytokines, or to kill Pseudomonas. So they're all very different, and I think as people develop antibodies, we need to move away from just developing IgG1s and think about how we can optimize those antibodies. And hopefully we'll have even more success than we've had to date. Thank you.
Thank you, Geoff. I love it when people talk about human germline V genes. It takes me back. Have you got some rules now as to which germline genes you should use and you shouldn't use? Do you go with the most popular, or?
Yeah, everybody, not really. Not really. We let the library decide. That's one thing we've tried not to do, is to pick a germline and try and graft onto a germline. We can't always tell just from the primary sequence. We like to use our screens, and we get what we get.
Right, but if you lock in your CDR3, if that's where you are, let's say H3,
it's a sub-region actually of CDR3.
Okay, so you're in H3. Can you basically shuffle back any V gene family? CDR1,
we've actually made chimeras between different families.
Yeah.
But then, which we liked as molecular biologists, but people don't like because they're not real, and they're not things that
if you're switching out all residues 1 to 94, it doesn't matter anyway. It's the whole V gene, right?
Yes, but people do like to stay within a family.
Okay.
VH3.
Okay, right. So keep it in the family.
So we try and keep it as uniform as possible. And of course, part of the library, you can make subset libraries for VH families, which we've also done.
I love it. I love it. Questions at the front first. We'll do it both. It's fine. We're cool.
Okay, so is this thing on? Yeah. So Jonah Rainey from MedImmune. Nice talk. And I'm curious for your Pseudomonas program, what the difference is between KB001 and KB001A?
Yes, KB001 was made in bacteria, and KB001A is made in CHO cells.
Okay, great. And have you looked for any synergy with antibiotics?
With
clinically between
with antibiotics?
KB001A, yes.
In animal models, yes, but not in the clinic.
Okay. Thank you very much.
Klaus Fuchs, Böhringer Ingelheim. For your anti-asthma antibody, do you need lung permeation, and did you measure lung permeation?
We did measure uptake into lung, as I said. So those individuals where we did see an improvement in FEV1, we did see a presence of the antibody. So yes, the epithelium is permeable where there's inflammation. So yes, we do see uptake into the lung, and we don't see the need to deliver directly into the lung by inhalation.
Okay, thank you.
And your KB001, I saw there's quite a nice dose response going on there. I'm just wondering if you're nervous about your 10 mg a kg. If you could go higher, couldn't you do more?
Funny you should say that. Yeah.
You can, presumably you can.
You can go higher, and there may well be plans to do that for one of the indications. Remember, one of the indications is chronic, and one is acute. So you might think for the acute, you want to give more. Chronic, it's a lot of antibody to give, becomes expensive.
We have a lot of antibody coursing around our veins, don't we? So relatively speaking, it's not a lot of antibody.
So we might stay at 10 mg for the chronic, but we may go high for the acute.
All right, thanks, Geoff. Great talk. Thank you, and thank everyone today.